Dear students, in this session we are going to discuss the events which takes place in light phase of photosynthesis. I already taught you the biochemical reactions which takes place during photosynthesis, during formation of glucose, broadly divided into two main reactions, that is first one light reaction, second one dark reaction. I already told you the light reaction aim is to generate assimilatory power in the form of ATP, NADPH2. The aim of light reaction is to synthesize ATP, NADPH2. It takes place in thylakoids or in grana. In dark reaction, the CO2 utilizes assimilatory power to reduce carbon dioxide to form the glucose. It is a summary of photosynthesis. So let us discuss the light reaction events in detail. Okay, the first is photophosphorylation. Photo refers light, phosphorylation refers formation of ATP, adenosine triphosphate. It is a bioenergy, the form of bioenergy. Okay, here the synthesis of ATP takes place in the presence of light. It is known as photophosphorylation. The synthesis of ATP in the presence of light is known as photophosphorylation. Okay, so before to knowing the details of photophosphorylation, we have to learn electron transport, which takes place in thylakoid membranes. In our last session, we have learnt about photosynthetic pigments. Pigments found at thylakoid membranes which harvest the light energy, ultimately converted to chemical energy. So in photosynthesis, in chloroplast, the photosynthetic pigments are organized into two main groups, namely photosystem 1, photosystem 2. So photosystem 1, photosystem 2. Pigments are organized into two groups named as photosystem 1, photosystem 2. So they receive a different kind of wavelength of light. The PS1 shows its reaction center chlorophyll A P700. It receives 700 nanometer wavelength of light. The PS2, photosystem 2, its reaction center chlorophyll A P680. It receives 680 nanometer wavelength of light. So the electron transport, the proton translocation, these are important events that act as driving force for photophosphorylation. So let us know the details about electron transport. In photosynthesis, electron transport occurs in two ways: non-cyclic electron transport and cyclic electron transport. So in non-cyclic electron transport, both photosystem 1, photosystem 2 are participated. Okay, so first the photosystem 1 receives higher wavelength of light. The photosystem 1 gets excited and expel a pair of powerful electrons. So these electrons reduce ferredoxin reducing substance. Ferredoxin reducing substance. From ferredoxin reducing substance, electrons reduce the NADP. So NADP transforms into NADPH2. So protons are here available from lumen of the thylakoid. Next, the photosystem 1 deficit is in oxidized state because it expels electrons. So the photosystem 1 restores its reduction state by receiving electrons from photosystem 2. So in photosystem 2, it receives 680 nanometer wavelength of light. It also gets excited, a released pair of electrons. This pair of electrons pass through electron transport system. So electron in non-cyclic electron transport system, pheophytin, plastoquinone, cytochrome B by F complex, plastocyanin. These are electron carrier proteins. They transfer the electrons in a downhill manner, in downhill movement. So when electron moves from pheophytin, quinone, cytochrome, plastocyanin, so they move and reduce the PS1. So the PS1 restores its reductive state, is a reduction state. Here PS2 is an oxidized state. It also differs from electrons. So in this condition, photolysis of water takes place. Photolysis of water takes place. So in thylakoid, the inner membrane possesses oxygen evolving complex. Oxygen evolving complex direct the split of water molecule in the presence of light. So due to split of water molecule, it releases a pair of electrons, a pair of protons and molecular oxygen. So these electrons moves to PS2. So PS2 restores its production state. So these protons moves either NADP or lumen of thylakoids. So molecular oxygen diffuses into external atmosphere. So listen attentively. While the electron moves from water molecule to PS2 electron transport system and PS1, and finally it reduces in ATP. So whenever electron takes place among photosystems, protons from stroma moves into lumen of thylakoid. In this case, cytochrome B by F complex, it plays key role. When electrons moves through electron transport system, cytochrome B by F complex absorbs protons from stroma. Again, they pumps protons into lumen. So these events ultimately cause photophosphorylation. We can discuss it in next session by explaining chemiosmotic theory. So we quick recap about non-cyclic photophosphorylation or non-cyclic electron transport. In non-cyclic electron transport, both two photosystems are involved. That is PS1, PS2 is involved. So both photosystems receives light energy, get excited, release the electrons, will finally reduce the NADP. So in non-cyclic electron transport, photolysis of water takes place. It is a water molecule oxidized. Water molecule act as a donor of electrons, protons and molecular oxygen. So it is a comprehensive detail of non-cyclic electron transport. So the second kind of electron transport is cyclic electron transport. In cyclic electron transport, only photosystem 1 is participated. So whenever the electron PS1 absorbs light and get excited and expel two pair of electrons, which moves through electron transport system and finally reaches return back to PS1. So in cyclic electron transport also, while electron moving through electron transport systems, protons moves from stroma to lumen. So these events form the base for synthesis of ATP. So it is the detail of electron transport. Dear students, in this session we are going to discuss chemiosmotic hypothesis. In our last session we have studied the events of light reaction. In photosynthesis, the entire biochemical reactions broadly divide into two prominent phases, that is light reaction and dark reaction. The ultimate aim of light reaction is to generate ATP and NADPH2. So the ultimate aim of light reaction is to generate assimilatory power, which is in the form of ATP, adenosine triphosphate, nicotinamide, adenine dinucleotide phosphate, reduced NADP. So the production of assimilatory power is associated with few biochemical events. So the biochemical changes that are result in the formation of assimilatory power. Already we learned in light phase, photosystems involved, they harvest light energy, gets excited, so they expel electrons. The electrons moves among photosystems. While electron transporting takes place, protons, the cytochrome pump receives protons from stroma. Same cytochrome B5 F complex pumps the received protons to the lumen. Okay, so here we can discuss in detail in photosynthesis or respiration. The formation of ATP is related with proton concentration gradient. Here Peter Mitchell, most important, Peter Mitchell proposed chemiosmotic hypothesis. According to chemiosmotic hypothesis, proton gradient is a driving force that causes formation of ATP, NADPH2. So how proton gradient is formed? Let us discuss in detail. First, it is a illustrating picture of thylakoid. It is a miniature pic of thylakoid. Thylakoid shows two membranes, outer membrane and inner membrane. I already told you the electron transport, whether it is cyclic or non-cyclic, both kind of electron transport takes place across the thylakoid membranes. Here, in non-cyclic electron transport, two photo systems are involved. So when these photosystems receive the light energy in respect to their wavelength, they expel electrons. So the electrons moves from PS to PS1. Finally, they reduce NADP to form NADPH2. So while electrons moving through electron transport system, cytochrome B/F complex absorbs protons from stroma. So this cytochrome B/F complex pumps protons into lumen. What is the lumen? The empty space, the space of thylakoid is known as lumen. Lumen is also known as proton reservoir. Lumen is the space of thylakoid that act as proton reservoir. Okay, so due to this, protons are accumulated in lumen via cytochrome B/F complex. Next, in light reaction, photolysis of water also takes place. When water molecule splits in the presence of light, in thylakoid, the inner membrane possesses oxygen evolving complex. So this oxygen evolving complex split the water molecule, which result in the formation of a release of electrons, protons, molecular oxygen. Here electrons moves to PS2, protons moves into lumen, molecular oxygen diffuses into atmosphere. So due to split of water molecule, hydrogens accumulated into lumen. So protons accumulated in lumen through cytochrome B by F complex and photolysis of water. So after these changes, proton gradient exists between thylakoid and stroma. The stroma shows low concentration of protons, whereas the lumen shows high concentration of protons. So according to absorption laws, the protons, ions moves from higher concentration to lower concentration. In this case also, the protons try to move from lumen to stroma, but thylakoid membranes exhibit impermeable to protons. So in this condition, the important enzyme, namely ATP synthetase, ATP, or you can say ATPase enzyme, it embedded in thylakoid membranes, it plays key role. So the ATPase enzyme shows two parts. The basal part is called FO. It acts as proton channel. It receives protons from lumen and pumps into stroma. Here the apical part of the ATPase is known as F1. F1 acts as catalytic site. So the FO part of ATPase enzyme pumps the protons from lumen to stroma. Whenever protons moves through ATPase, some amount of energy is released. The released energy utilized by ADP and it fixes inorganic phosphate to form the ATP, that is phosphorylation. So this theory states that proton gradient is the driving force for the formation of ATP. So the chemiosmotic theory applicable in photosynthesis as well as respiration. So in the photosynthesis or respiration consider the most important biochemical reactions. So in both cases, the ATP formed, synthesized ATP is known as cell currency, the bioenergy. So ATP formation takes place according to chemiosmotic hypothesis.